So to review the LAC operon, it consists of one promoter that tracks the RNA polymerase, an on-off switch that can be affected by an operator, and the genes that are controlled by the entire operon. Here's the full picture. Okay, so here we have the repressor being produced upstream. This is referred to as upstream, and this is regularly referred to as downstream. Okay, so upstream, the repressor itself is produced. In this instance, when lactose is in this environment, so we have an E. coli bacteria that is experiencing an increase in lactose in its surroundings, okay, it's going to need to digest that lactose. So the lactose itself affects the repressor, changing its shape. Once its shape is changed, it can no longer attach to the DNA at that spot. So it is now disconnected from the operon site, okay, from that operator site. Now the RNA polymerase, as you can see, is free to move downstream, producing all three of these enzymes. These three enzymes then have a function in digesting lactose. So the lactose present turns the operator on, okay, by changing the conformation of that repressor. So lactose binding to the repressor, changing the protein conformation, and the operon with no repressor allows that RNA polymerase to bind to the promoter and produce the RNA transcripts that are needed to make those three enzymes. Again, we're going to skip the videos that are in this Prezi. You're welcome to use the link to the Prezi to watch those on your own. Okay, so we have three genes that are indicated as LAC-Z, LAC-Y, and LAC-A. LAC-Z produces beta-galactosidase. Its purpose is to break down the lactose. It's going to take that disaccharide, split it into two. LAC-Y produces a permease, which is a membrane-bound enzyme that provides a channel for more lactose to be diffused into the cell. And LAC-A produces a transacetylase. There are some arguments as to exactly what transacetylase does. However, in the resources that I've been able to dig into, the most commonly referred to function of the transacetylase is that it has some detoxifying, some, if you will, cleanup job for metabolizing anything that's left over from this process. Okay? But certainly the LAC-Z and LAC-Y enzymes have much more definitive roles in the process. So that is a positive control. Okay? The LAC operon is a positive control. Next, we're going to take a look at a negative control. Okay? The TRP operon is also in the E. coli, and we're going to take a look at that next.